In the years following the Second World War, a scientific mystery captivated researchers. How do plants transform sunlight and air into the sustenance of life? This question led the American chemist Melvin Calvin on a groundbreaking journey into the heart of photosynthesis. Calvin's team at the University of California, Berkeley employed a novel tool, radioactive carbon-14. By introducing this tracer into algae, they could track the path of carbon dioxide as it was captured from the air and transformed into the building blocks of life. Their chosen subject, the single-celled algae chlorella, proved ideal for this task. Simple to cultivate and quick to reproduce, chlorella provided ample material for analysis, ultimately revealing the intricate steps of carbon fixation. The groundbreaking research of Calvin and his team unveiled a cyclical process, now aptly named the Calvin cycle. This intricate dance of chemical reactions takes place within specialized compartments in plant cells called chloroplasts. The cycle begins with the enzyme Rubisco, the most abundant protein on Earth. Rubisco captures carbon dioxide from the atmosphere and fixes it onto a sugar molecule, thus initiating the cycle. Through a series of energy-intensive steps, this molecule is transformed, ultimately yielding the energy-rich sugars that sustain the plant and, by extension, all life on Earth. The Calvin cycle is a continuous process, constantly replenishing its starting materials to ensure a steady supply of sugars. These sugars not only fuel the plant's growth and reproduction, but also serve as the foundation for all other organic molecules, forming the very fabric of life itself. Central to the Calvin cycle is the enzyme Rubisco, a molecular gatekeeper with a critical flaw. While its primary role is to capture carbon dioxide, Rubisco can also bind to oxygen, a quirk with significant consequences. This tendency leads to a process called photorespiration, a wasteful side reaction that consumes oxygen, releases carbon dioxide, and drains the plant's energy reserves. Photorespiration is particularly problematic in hot and dry environments where plants close their pores to conserve water, limiting carbon dioxide intake and increasing oxygen levels within the leaf. Photorespiration can significantly reduce a plant's photosynthetic efficiency, impacting growth and yield. However, nature, ever resourceful, has provided certain plants with ingenious adaptations to overcome this challenge. Over millions of years, plants have evolved diverse strategies to thrive in various environments. C4 and CAM plants, for instance, have developed remarkable adaptations to minimize photorespiration and conserve water, particularly in hot and arid regions. C4 plants, such as sugarcane and corn, possess a specialized anatomical and biochemical pathway that concentrates carbon dioxide around Rubisco. This mechanism reduces Rubisco's exposure to oxygen, thereby minimizing photorespiration and enhancing photosynthetic efficiency. CAM plants like cacti and succulents employ a different tactic. They open their pores at night to capture carbon dioxide, storing it in a form that can be used for photosynthesis during the day when their pores remain closed. This strategy minimizes water loss while ensuring a steady supply of carbon dioxide for the Calvin cycle. These adaptations highlight the remarkable plasticity of life, showcasing nature's ingenuity in overcoming challenges and thriving in even the harshest environments.